Okay, I'm going to give another example of what we refer to as receptor regulation. Okay, let's say we introduce a drug that in this case is not an agonist, right, where it would act like the neurotransmitter and stimulate those receptors as a neurotransmitter would, right? But let's say it's an antagonist. So it'll bind to the receptors that are present here, right, and the postsynaptic membrane, postsynaptic neuron, and it'll block their function. It'll prevent the neurotransmitter from stimulating, and it will stimulating them, and it will prevent them from, releasing the G proteins and opening the ionotropic, channels, right, to allow for changes in membrane potential. So if this is true, you have a lot of antagonist exposure, you're going to get fewer G proteins, right? And then you're going to get fewer second messengers, right? This is going to be noted by the nucleus. There's going to be a change. There aren't going to be nearly as many chemicals coming in here to stimulate, both inhibit and stimulate production of particular, genes, right, and then proteins. So you're going to get changes in gene expression. You're going to get changes in the synthesis of receptor proteins. And one thing that can happen with, a lot of antagonist exposure is you might get increased production of certain receptors. It's almost as if, the signal is being blocked here. So in an attempt to sort of compensate, the cell adds more receivers, right? Adds more receptors in the postsynaptic membrane. This is referred to as upregulation. This is an increase in the number of receptors and the sensitivity of the postsynaptic membrane in response to both drug and neurotransmitter. So to put these together, you can have agonist exposure or antagonist exposure, drugs, right, that can be influencing receptors, and because of their influence at those receptors, because they change the number of, the concentration of G proteins that are released, and they change, the message that is received by the genetic material, and they change gene expression and receptor protein synthesis, you get compensatory changes in your neural networks. The neural networks literally change the strength of their connections, the number of their connections, right? The sensitivity of their connections to drugs. Actually, so the next time a drug is introduced, there may be downregulation that has occurred on these synapses where it acts, and you might not get the same response, from the same level of drug that you previously were exposed to. It may no longer stimulate these neurons in the same way, right? So this underlies the development of what we refer to as tolerance, right? For example, a great example would be a drug that stimulates dopamine receptors, and we'll talk about dopamine receptors in more detail. But maybe, dopamine receptors, we will learn if they're stimulated, it usually feels quite good. And so, you want more of that drug. But if you introduced a great deal of it and you stimulated those dopamine receptors, right? And then you get, fewer, different receptors expressed at those postsynaptic membranes. The next time you take this drug, there's not nearly as much, protein. There aren't nearly as many receptors at which this drug may act. So you do not get the same level of stimulation. You'll need to take more of the drug. So this underlies what we refer to as tolerance in exposure to drugs.